Okay, let's get started. Welcome to the MIT Colloquium. Today it's a great pleasure to introduce our own colleague, Professor Ming Guo. Ming told me to get the introduction very short. He has lots of content to show you. So I'll make it brief. Ming is currently associate professor at the Department of Mechanical Engineering at MIT. His group works on developing tools to characterize and understand the cells and tissues as soft active matter. Before joining MIT, Ming obtained his PhD and a master from Harvard and his bachelor from Tsinghua University. Ming has won numerous awards. I'll just mention two: Alfred Sloan Fellow in Physics and the IUPAP Young Scientist Prize. Ming currently is the associate editor of the Journal of Biological Physics. Today, he will talk about pushing multicellular living systems to extreme reality and the virtual world.
All right. Thank you. Thank you, Shenhe, for the introduction. And thank you all for coming. So earlier today, I realized I might have made a mistake when I put up this title, and someone asked me this morning for a Zoom link. And I said, there's no Zoom link, it's in person. And he was like, oh, that's what you mean by virtual world. It's not on Zoom. Anyway, so let's get started. So we all know that cells are known as fundamental building blocks of life, right? And for each adult of us, we have about 37 trillions of cells and about a couple hundred different types of these cells, and constantly are organizing, moving, jiggling in our body. It is a self-organization and the spatial and temporal coordination of the properties of these cells and their positions that allow us to be a functional unit that can stand here and talk to all of you. And my group has been very interested in developing tools, but also to really understand what's going on in the system. So what are some of the fundamental rules that regulate these behaviors? This include a characterization of individual cell mechanics. As you can see here, I think I'm out of battery, so I would just directly point it to you. And also it includes the mechanics of the extracellular matrix. And then more recently, we start to work on multicellular systems. So that's what I'm going to tell you about today. So I'll use the time to tell you two major parts. So one is the experimental work that we have been using multicellular living systems to characterize the spatial temporal cellular behavior in several different systems. And for the second part, I really hope to convince you that we should really start to use AI to understand these systems. And one of the methods that we promote is a graph neural network that I will talk about at the end, okay? All right, so the very first couple of years of my group at MIT, we have been primarily focused on individual cells. So we're interested in what cytoskeletal component are constructing the cell, how they are regulated in terms of the composing the cell, setting their mechanical properties. Here is one cell that you can see is a fixed cell on a glass cover slip on 2D. And in this cytoskeleton, what I'm highlighting here is the three major components. The pink here is F-actin. They're important for cell adhesion, cell migration, contractility. They're really well studied. And the second type in yellow here is microtubules. They are really important in transporting the cell. But there's really a third type called intermediate filament, which is rather underexplored as a frontier, even as of now. So they sort of become more and more popular in the last 10 years. But some of the fundamental questions, for example, why are they there in the cell with their functions, are still unclear, okay? But what people do know is intermediate filament corresponds to more than 70 different human diseases, and most of these diseases are inherited diseases that can carry it over from parents to kids. But why this is the case is unclear, okay? And what we're proposing is their main role might be simply structure and mechanics. And collaborating with Shenhe's group a couple years ago, we proposed we might just understand a cytoskeleton as an interpenetrating gel, that we have intermediate filament, which is very floppy but very stretchy. In the meantime, we have actin and microtubules that they're rather fragile, but they can self-heal. So when you combine them together, you can profoundly enhance the toughness, stretchability of the cytoskeleton. So in a very recent perspective that we put together with a few pioneers in the field, we really start to talk about this picture. So even as of now, the research about intermediate filament is very underfunded at NIH. But last month, a few of the program managers starts to show this paper in their progress report. So what I'm hoping for is it starts to gain more and more attention at NIH. Therefore, more people can work on this together. All right. So talking about the work in my group, we're still very interested in IFs. This video is what Mary took, I think, last week. That you can see the green intermediate filament are kind of covering the individual cell nucleus. And as the cell nucleus rotate, it can tweak the intermediate filament a little bit. So the objective that we're trying to look into is whether intermediate filament here provide a mechanical protection for the nucleus and their genome stability. And what Camille has been working on is further investigated how intermediate filament is involved in collective cell migration on 2D and also in 3D. All right, so for today, I will focus on multicellular systems. And the reason is simple. So all the nice images you see are growing cells on a cover slip, right? That's not their natural state. If you take cell out, grow them simply on glass, they're not going to behave the same way as they do in the body. Just imagine it's a nice day outside, right? We don't wear too much, but if I move everyone here to the North Pole and we do the seminar there, I'm sure we're not going to feel the same way as what we're feeling here, right? That's exactly what's happening to cells. In the tissue, cells are, surrounded by other neighbors. They constantly talk to each other, and they're surrounded by extracellular matrix. To study their behavior, we need to put them back to their native state. So over the years, we have developed a number of different systems. This is one of the systems pioneered by a previous postdoc, Yulong Han. And what he did is he grew an individual breast epithelial cell in 3D hydrogel. And by tuning the hydrogel stiffness, he can make the cluster to start invade, just like what breast cancer does, as you can see. So each fluorescent blob there is an individual cell. It's a 3D video projected on 2D, okay? By imaging the system, he can measure the size of the cell, the migration speed of the cell, and many other characters. And he can also use optical tweezers to go through this cluster, each cell one at a time throughout this development process over 2 weeks. And what he found is very interesting that he see the core of that cluster have smaller cells, and cells are also stiffer. And if you move away to the boundary of the cluster, the cells become larger and also softer, and also migrate faster. And these characters seem to be quite relevant in driving the invasion process of this cluster. And why this happens is because of water transport within this tissue. So basically, the tumor stress compress the water to flow from the center to the outside to swell the cells on the boundary. So we really highlight the importance of the intra-tissue water in this study. And then another grad student in my group did a follow-up work, basically asked the question, how is the liquid in the tissue influence the whole tissue level mechanical properties? And he built a very interesting device using a high position microbalance with a step motor to compress this 100 to 200 micron diameter tumor cluster or a microtissue, in a sense, to measure their force displacement curve. And by doing that, he basically showed the intercellular liquid flow is actually dominating the poroelastic behavior of this cluster. In the past, people have always been talking about viscoelasticity of tissues, but essentially ignore the water part in this, and which Fan now show that is a really important factor that we should think about, okay? And currently, we're collaborating with the Roger Camp's group, with Shun Jackie, to study the pore-elastic flow in the brain, which is highly relevant to a number of brain-related functional and disease processes. All right. So 2 weeks ago, if some of you come to Ritu's talk, you must have heard that mechanical force is important for tissue function. It can change the biology of neurons and the muscle and neuron connections. The question we're asking here is what about the mechanical properties of the cells themselves? Because the cells are fundamental building blocks, right? If we change the property of these blocks, do that matter? And some of the beautiful work by a previous postdoc, Yue Li, are on single cells. So he basically takes individual cells and change their mechanical properties. And he found this profoundly changed gene expression, stem cell fate, and some of the important signaling that can drive different diseases in the embryonic development process. And we propose changing mechanics of the cells as a way to engineer living systems. But my question is, what happens in a more complex environment, right? For example, in a developing embryo, what can we see? And this is a work by Haiqian and Lani in my group. And we focus on mouse pre-implantation embryo development process. So they start from one single cell. At about 4 or 5 days, they develop into about 128 cells or 256 cells, and then they're going to implant. So we focus on the process before they implant. And using some of the techniques that we developed, we can map the rheology of each individual cell throughout this entire embryo during the entire embryo development process, just one by one at different time, while we can keep the embryo alive. And after we finish all the measurement, we fix the embryo and send it to our collaborators, Fei Chen's lab at Broad Institute, that they can do in situ mRNA sequencing to start counting how many a specific type of mRNA each cell has. So the way that they're doing is by imaging. So mRNA are produced by the cells, and the next step is to generate proteins, right? And if you know mRNA that you're interested in, you can design a complementary DNA that has a fluorescent tag, and then you can flow this DNA in to tag your mRNA of interest, and then do fluorescent imaging that you can see it, right? And if the density of the mRNA is not too high, you can even distinguish them one by one. So each round, we basically tag 4 different colors that allow us to use this imaging-based method to count 4 types of mRNAs. And after we're done with this round, we photo bleach all of them and then do another new round of 4 new ones. And you can do this over and over. In the end, we achieved a whole embryo imaging-based mRNA sequencing, and we can image up to 150 different genes. So in this intact embryo. So that's pretty amazing. And here you can see this is one of the example videos. Each fluorescent speckle is one mRNA, as you can see. So you see them starts to come up. This is the Z stack. We're moving from the bottom up. And imagine if I stain the cell membrane in the sample, I can really start to count how many of this type of mRNA in this cell. And I can take that to cross-correlate with my cell mechanics measurement to see how they're influencing each other. So here are some of the example embryos that we imaged, including the nucleus. In the bright color here, and the green highlights the cell membrane. And just to show you that we can really count them one by one, okay? And briefly, if I now cross-compare, the x-axis is the compliance of the cell, and the y-axis is the expression level of a particular gene of interest. You can see that they're fully correlated in a very interesting way. So the more compliant cells have a higher expression level of the stem cell markers. So stem cells seem to be softer in this embryo. And if you look at the stiffer cells, and they're anti-correlated with the stem cell markers, but actually correlated with some other genetic markers associated with the trifectoderm development, which is the outer layer of the embryo. So overall, we found a very strong correlation between the cell mechanical property and the gene expression of many, many different genes. So I was hoping to see a causal relation. For example, cell mechanics will lead, and then I see the gene expression, which is not what I saw here. So the next step, I think, is we're going to massage the embryo in a different way to see if we can break this correlation to test it. All right. So in all the samples or different systems that I showed so far, cells are never staying there still. They're always migrating, right? In our body, the trillions of cells are constantly jiggling and migrating. This is actually really important. If I make a scratch on my skin, cells have to migrate over to regenerate this broken tissue, right? So cell migration is definitely crucial. And because it's really important, it has been studied for many years. Here I show two classic examples. One is from one of the pioneers in this field, Jeff Frenberg's lab, that they grow MDCK cells on 2D surface and then look at their collective migration. And on the right-hand side is another pioneer, Xavi Tripat's lab, that they study collective migration in wound healing and how they use the substrate stiffness gradient to guide their migration. Okay, most of collective migration studies are actually done on 2D flat surface. But I also, in the meantime, want to remind you, if you think about our tissue surface, they're never flat on 2D. They're actually always curved, right? And a couple examples, the long alveolar sphere, the embryo, and also some of the cryptid structures in our intestine, the curvature radius is on the order of a couple hundred microns. So they're never flat. And the question is, how do these curvature influence the collective cell migration? So is that really fair to do everything on 2D, or should we start to think about 3D curved surface, right? This work is done by previous PhD student Wen Hui. Here, what she did is she starts to fabricate the wells and also the dome structures of isotropic curvature with different curvature values using PDMS. And then she grew a layer of MDCK cells here on the surface. And because the cells have a fluorescent nuclei, she can start to track the cells and follow their trajectory and starts to analyze it. And a couple curvature values I should mention with a curvature radius 125, 225, and 325. So, both positive and negative, but both are isotropic. So the first thing she did is to calculate the velocity direction cross correlation. We ignore the amplitude of the velocity here, but only consider the direction, right? And to calculate the cross correlation, we actually move the two cells at different locations along the geodesic length to the same point, and then compare their correlation in terms of the angle. Imagine if a whole sphere is spinning and I have two cells on the two poles. After I move them over, they're going to be aligned, right? They're fully correlated. So using this way, you can test the correlation. And here is a typical correlation function on the flat surface. You can see initially they're fully correlated with themselves. Here's the distance between cells, and then gradually the correlation decay, okay? And if I do the same calculation in the curved surface, clearly you can see the correlation curve is being pushed to the left, so the decay is faster. And if I keep increasing the curvature, I see a more and more clear trend that the decay is being faster and faster. This really suggests that there's some swirling patterns in the cell packs, that cells on the two sides of the swirl are actually anti-correlated, as you can see here. And we're interested in taking the correlation length. So a very easy way is to take the correlation length from the minimum of this correlation function. And by doing that, you can see the correlation length basically decay as the curvature increase in both directions, right? No matter positive or negative. This is quite interesting. It basically suggests as long as the surface is curved, they change their migration dynamics. They don't seem to care if it's concave or convex, in our case at least. So this results clearly indicate that the correlation length is different, and which means cells are less collective on the larger curvatures. They're more chaotic, right, with shorter correlation length. And another thing that she did is perform particle imaging velocimetry based on the cell locations, and then she has the velocity field of the migratory cell packs. And then she can basically dissect this velocity field into three components: divergence, curl, and the shear. Divergence being swelling and shrinking. Curl is a swirling pattern, and shear is if I have two streams moving past each other, I have a strong shear. And then we can basically analyze the three components one at a time. So what I'm showing you here is only the divergence field. The color represents the amplitude of the divergence. And you can clearly see on the flat surface, everything seems to be more calm, right? So you have some large patterns, but very, very small amplitude. As I move to the higher curvature side, you see the color become more and more intense, and the pattern reduce their size, suggesting, it's really more chaotic. All the divergence field become more intense. However, the pack size is reducing also. And you can see this more clearly from this probability density function. On the flat surface, everything is near zero, and as I increase the curvature in both directions, they spread out more, the values, suggesting it's a lot more chaotic in terms of the flow field here, okay? So this results again confirm that the multicellular flow is indeed more dynamic, and the flow pattern is smaller on larger curvatures. So there's a very clear curvature dependence. So all of this are still on PDMS well. So it's indeed semi-2D or semi-3D, but it is not in the real environment. So we have always been wondering what happens if I have a real system. So do curvatures still matter, right? To do that, we start to collaborate with Dr. Derek Colton at Boston Medical Center. His group is pioneered in taking skin cells, reverse engineer to produce induced pluripotent stem cells, and then forward engineer them into the person's lung alveolar structure. And he has stem cells from both healthy person and diseased patient to understand the specific reason underlying a certain type of lung disease. So here we're just using his healthy samples, okay? So all of these are lung alveolospheres that his group developed, and they are essentially a balloon embedded in three-dimensional gels, okay? They're fully in 3D and only a single layer of cells with liquid inside. So you can see the cells are constantly migrating on the surface. It's very active. And Weihui obviously can apply all her analysis tools on these systems as the alveolospheres grow larger and larger. And what she saw is consistent with the PDMS well. So now what we're showing is divergence, curl, and the shear field. As the alveolosphere grow larger and larger, you clearly see the calm down of the all the three field very clearly, right? So all of these, again, confirm the flow is more dynamic on larger curvatures, right? As the alveolosphere grow larger and larger, everything seems to be more calm and more correlated in a way. So why this is happening? If you take a cell monolayer, because they're so crowded, I'm always sharing an edge with my neighbor. Therefore, often the cells look just like polygons. And a very typical polygon shapes are actually hexagons. And we're collaborating with Max B's group, who's an expert in simulating 2D cell monolayer and their migration. And according to him, hexagons are a way to reduce the phrenology, okay? And when you have a number of the hexagons, they actually like to cluster together to form packs. And then we went back to check our experiments. Indeed, you see the blue color hexagons, they indeed form clusters in our experiments. Very interestingly, I don't quite understand still, but I think this is pretty cool. And how do we understand it? If you think hexagons form more stable packs, which we can examine in our experiment, when they form a pack, they stay together for quite some long time. And that's sort of similar to colloidal crystals in a way. So just tell me what I mean by this. So if you take a bunch of plastic particles which have attractive interactions or weak attractive interactions, and you put these particles on the surface of an oil droplet, and because the particles are attractive, they like to attract and form a crystal, right? And typically they will form a flat 2D crystal, but right now they are embedded on this curved surface. So they have no option but bend themselves, right? And when they do that, they have to increase their elastic bending energy, which is a problem. To avoid that, they break into smaller pieces, right? And obviously you can write out an equation to calculate the free energy change. The first term is the energy gain associated with the forming cluster. The second one is the energy penalty associated with bending them onto this curved surface. And if you take derivative of the free energy, you can find that there's a critical pack size that these hexagons can form, which is linear with the curvature radius in this case, okay? And if your pack is larger than that, the bending energy will dominate to break them apart into smaller pieces. And to check if this is what happens in the experiments, we directly measure the size of these hexagon packs. And indeed, we find it is linear with the curvature radius. Very surprisingly when I saw this. So I still want to say this is a working hypothesis. And the main reason is because we don't really know if there is indeed an energy gain when the hexagons form packs. We can't really measure it. And we also have not directly measured the bending behavior, whether the packs can bear bending to some degree. We don't really know. So if some of you have good ideas, I would love to hear more about this. All right. So besides the collective migratory behavior, Wenhui did some other analysis to look at the topological packing behavior of cells on the curved surface. And she found that not only the hexagons form packs, you actually also have more hexagons when the alveolospheres grow larger. So I'll skip this part and just tell you the conclusion we have. It seems the substrate curvature really significantly changed the collective submigration and also the collective subpacking. So that's a strong suggestion that when people consider collective submigration, you can't just do it on 2D surface. We must start to consider 3D curvatures and those effects. Okay. So here is, I think Wenhui submitted a plot and it's been selected to print on the APS t-shirt last year. She was very happy about that. I have one. All right. Okay, so for the remaining time, I want to fully jump into the virtual world to tell you about how we use graph neural network to start to predictively model a multicellular process. I think this is really cool. For a number of years, I have been very skeptical about this using machine learning. And Haiqian is the one that keep pushing me to think more and more about this. So I think this is all Haiqian and also Shaoxi's credit. Is amazing work from them. I'm just a co-pilot that occasionally gives some random suggestion that can only delay their progress. So you should ask them. All right. But even I have been skeptical. I think the idea of a graph structure is something we have been thinking about for years. This is the first project that Haiqian worked on in my lab. It was during the COVID time, so he can't come to the lab. So we're thinking what to do remotely, right? And then we came down to thinking about this problem. So as you see in this video, I have a selective number of dilute MDCK cells, and then they divide until they fill the entire space. They're still migratory, and then keep dividing, increasing the density until a point that they reach a process called a jamming transition, that suddenly the whole pack freeze, that they can't move anymore. It's been a popular topic in the last 10 years in the field of biophysics. And the question we have is, can we propose and some spatial order parameter to describe the system? can these other parameters be used to identify some of these transition process, basically, right? It's a very simple question. And in the field, there are some existing attempts. For example, cell aspect ratio or shape index being the parameter divided by the square root of the area. All of these are based on individual cell character, right? What we want to do is to see if we can do this in a broader collective picture. In collaborating with Rohan, that Haiqian did something I think really cool, is first to connect each individual cell nuclei by lines, and then this way you can triangulate this entire space, right? This is called a diagonal triangulation. Take the 2D picture as an example, but it can do everything in 3D actually. So if you triangulate this space on 2D, and you can define each neighborhood by 3 cells as a triangle, right? And then you can start to calculate the deformation gradient that each triangle undergo as compared to a perfect control frame being the equilateral triangle, okay? And once you have the deformation gradient, you can further split it into 2 types of deformations. One is a dilation and a shrinking. The other is a distortion of the triangle. So we have the volume and the shear parameter here, okay? And that you can see for a graph like this, we can plot each triangle, calculated there are 2 parameters, and put a point on that graph. In the end, I will have a pointed cloud as you see here. And using that, we can define a configurational fingerprint that capture the current state of this entire multi-cell systems, okay? To make it simpler, we further synthesize all these data to define 2 relatively simpler ordered parameters. One is the volumetric order, which we capture the variance of the volume fluctuation. And the other is a shear order parameter. We simply just use the average value of the shear distortion order. And by using these 2 order parameters, we can start to quantify what happens in different systems at different times. Again, take this growing 2D model layer as an example. We know there are 2 transitions. So one is from subconfluent to a point that they are all connected to form a confluent layer. And then there's another transition is when they start to jam, then they stop migrating, right? There are 2 transitions. And using this order parameter, you can see we first see the volume order parameter reduced to a point that they stop changing. That suggests that the density fluctuation is saturated. So that defines the confluent, non-confluent to confluent transition. And the second one is if you look at the shear order parameter at a later time, the shear distortions stop changing as well. That suggests that the system starts to gain some rigidity, right? So that's the jamming transition. So using the volume as shear order parameter, you can successfully describe and capture different stages in this very simple system, okay? And here's another quick application that we applied to a fruit fly embryo that you can see as the imagination happens, the 2 order parameter both suddenly starts to diverge. Once we publish this, it appears to be quite popular. A lot of people are using it now. Just give you 2 examples. One is by Karen Keza's lab at Columbia University that they use the volume as shear order parameter to capture different developmental stages in the Drosophila embryo. And they even use it to identify if you genetically mutate the embryo, what can you see? So by using our order parameter, it's been very successful in their study. And on the right-hand side is another recent work from O.V. Charter's lab at Stanford. They used the volume and shear order parameter to define different parts in the breast cancer invasion. So it seemed to be very useful. That I'm sure Haiqian is very happy about it. All right. So come back to more data. Here are some of the experimental systems that we have in the lab. I only talked a few selection of them, but we have a lot more. And my point really is identifying the order parameter. It relies on our understanding of the system, right? You have to have a good intuition to identify what parameters are important, and then you do it. Sometimes you did it right, but most often you did it wrong, right? It doesn't help. When you have this much data, relying on intuition, I think is very difficult. That's why I think AI is very natural in this process. Actually, as I look at this data here, I feel I see more of it on my own screen. So maybe we should start looking at them here. Anyway, all right. So to use the AI or machine learning, the very first question is, how do we even digitize the data, right? How do I convert these videos into a format that machines can use? And the next question, obviously, is how do we predictively model this process, right? The ultimate goal is to build a foundational model that allows to really predict what's going to happen when you have a perturbation, when you stress it, all different scenarios. So let's first discuss the data structure. The most traditional way, obviously, we have the video here, right? We know video are connected by images, and each images is composed of the individual pixels, right? If you take the intensity of each individual pixel, then on 2D or for each voxel in 3D, you have the data, right? That seems to be quite successful in some imaging identification process for histology, for example, but that doesn't really capture the fundamental process that we have in mind. For example, tissue is built with cells, right? How do we present that picture? And one way to do that is to use the graph representation. It's actually very natural, I think. A graph includes nodes and edge, right? The nodes are individual components that can include, you can add on node identities and some of the node-related features. And the edge are simply lines connecting those two nodes, right? And between each pair of nodes, you have some connections, and that information can be added to the edge information. To make it a little more intuitive, here is a graph representation for main characters in Harry Potter in the Chamber of Secrets that you see some of these characters, right? Their names are labeled with color. That's basically a node property, right? It represents which house they're from. And you can add multiple different properties. For example, are they from a Muggle family or not, or things like that, right? And you can also have the edge information as the interaction between each of them. And that's essentially what we can think about cells, right? Each cell is a character in this book, and between each cell, there may be some interactions. And that's the information you put on the edge, right? So it's very naturally, I think, using graph to think about this multicellular process. And there are actually, two major types of mechanistic pictures in our field that are sort of related to this graph identification, actually. So on the left-hand side, is the first major type is the colloidal wheel that people typically think about a multi-cell system as a colloidal system. As an example, what I showed you earlier is basically by treating each cell as a colloidal particle, and we try to understand how they pack, right? So that's a classic colloidal wheel of multicellular living systems. And in this very classic paper by Jeff Fredberg and Dave Waite's group, they show multicellular collective cells can even look like a colloidal glass system. So it's a very typical colloidal wheel. But on the other hand, a lot of people will suggest that we should view tissue as foams, right? This dated as back 100 years ago to Darcy Thompson in The Growth and the Form. That when you start to think about the membrane tension, tissues should be thought as the foams. And there's some recent papers as well. So which is a better descriptor of tissues has been debated in the field. And what I want to propose here is it's actually not a problem at all if you think about a graph structure. For example, we can have a dual graph. That one graph represents the colloidal wheel. Each node represent a cell, right? And the edge is the cell-cell interaction. It can be elastic repulsion, it can be adhesion of different types, and you can have a separate graph structure for the foam wheel. And in this case, a node is not a cell anymore, but actually a vertex in that case. And the edge is the actual cell edge. And then you can have the membrane tension information, all different actual cell boundary information on it. And once you have the two graphs, you can combine them into a dual graph and do machine learning with that. So it's very easily just combine the debate into one unified picture now, okay? All right, so let's see how it works. So the first task I want to talk about is using graph neural network, can we predict the cell migration speed from a static snapshot? And that's a major question in our field. Just imagine, if I can take a biopsy, I take a picture of the cells, I immediately know how quickly cells migrate in the body, then it's profoundly useful in cancer diagnosis or you know, some other process, right? Therefore, a lot of people have been working on this. We're going to show that using graph neural networks, it can make almost a perfect prediction. So I'll skip the details of the model, but the experimental flow is we basically generate about 200-ish videos like this, and we split them, use about 200 at the training side, and then use the remaining 20-something at the testing side. And we can embed a different node and edge features onto the model, right? Just show you the results here. The x-axis is the mobility of the truth that we directly measure from the video. The y-axis here is the predicted value. You can see they're perfectly correlated, right? So suggesting taking a snapshot, I can really predict the average migration speed of the cells in this data set now, okay? And how does that compare to some of the current method? The current best method is based on the cell shapes, as I mentioned earlier. If you take the shape index and plot it against the migration speed, they're still correlated, but you can see the correlation is not nearly as good as what the neural network can do, right? Is at this point, I think I was fully convinced by Haiqian that this is really a great idea. But another question is why this is the case, right? Obviously, machine learning can do pretty good, but what exactly did it pick up? Can I learn from it, right? What is the physical parameters? And that's an ongoing project that we're actively working on. Shaoxun is spending a lot of time thinking about this. But just give you a very quick one of the example. For example, you can do ablation tests. It's not real ablation, but you can selectively ablate some information from your data set or from the neural network to hide it from the model, right? And see how it does. For example, the full model we have our area and the parameter and the edge information, right? Everything is considered. I can get rid of the edge information, only have the node information. And you can see the prediction is worse than the full model, right? And I can also get rid of the node information and only use the edge information. Again, you see the performance is even worse. So all of this are suggesting some of these parameters being ablated are playing an important role. I don't really have a quantitative expression for any of these, but at least qualitatively, I think this can be a good way to help us to identify important parameters that the machine learning algorithm is making a good prediction, okay? All right, the next major goal for us is, we obviously can predict the average cell migration speed, but can we predict the individual cell behavior also? I have this Drosophila embryo development process. Can I tell what each cell will do in the next minute, 10 minutes, 20 minutes, or half an hour, okay? And to do that, we collaborate with Tomer Stern's lab at U Michigan, and their expertise is to use laser light sheet to take really beautiful full embryo development process videos. And it's highly complicated that their entire lab only has 4 of these. So we just use these 4. It'd be great to have more, but currently we can't find more. So this is one of the embryos, and all the cells are segmented already that you can see how things are happening. And it's about an hour for this video, the length of the video. To be brief, we apply the dual graph idea onto this. So this is a dual graph that Haijian generated based on the this embryo videos. And then he feed the dual graph into a graph encoder and then split them according to what specific task they want to do. For make an edge prediction, node prediction, or a global prediction, right? And the first task we did here is to predict in the next minute in future which cell pair will lose their connection. So I have a pair of cells share an edge, right? At some point, they're going to lose their shared edge. And I want to predict in the future 1 minute which pair will lose it. And you can see that the prediction based on the model is labeled by the black dots, and the ground truth are labeled by colors. The blue color labeled the cell pair that don't lose disjunction. The pink ones highlight the cells that do lose their junction in the next minute, right? You can see they really overlap quite a while. Obviously, the video is too fast, so I just show you a few snapshot, and the 4 different views are, from the front, back, and top down, I think, showing the same embryo. That you can see the black dots really co-localize with the pink cells quite well. Overall, I think we have about 90% accuracy in terms of this prediction. I think is really cool that we can tell in the next minute with 90% accuracy what's going to happen. You can, again, perform a Bayesian study to tell which parameter might or might not be important in this. I'll skip this detail to tell you one last thing is the previous task. We basically make a prediction is in the next minute whether a certain thing will happen, right? The next question is, can I even know when it's going to happen, in 7 minutes or 8 minutes, right? So to do that, we're not only looking at the loss of cell-cell junction anymore, because there are different scenarios that can cause a loss of junction. We split it into 3 subtypes. The first one is the imagination. Some cells suddenly starts to fold into the embryo, and that's the way that they lose their junction. And the second one is some cells starts to divide over time, right? When they divide, they also change their junction. The third one is the actual neighbor swapping that happen while this embryo is developing. And all these are experimental data with the color marking the precise time when a certain event will happen. And how does that compare with our models? Again, I will skip the details. Essentially, we split the encoder into 3 different halves for different tasks because each task has a slightly different requirement. And here is the results. So for all the 3 types, the x-axis marks the exact time that each cell undergo each process, right? And the y-axis is the predicted value. You can see for all the 3 tasks, it's fairly well, suggesting we can really make a prediction with a fairly good confidence that what will happen to which cell and when, right? I think I was very amazed by this, and I want to know more about this algorithm, what exactly the model really learned. I think that's a long list of, things I want to know. Okay, I'll skip this and just tell you the last thing, which I have to, since I asked Shaoxun to prepare this video for me, I have to show it. So, currently we're collaborating with Merton's lab to see, using this graph neural network, can we start to learn, morphogenic lineage? If I genetically perturb the system, can the model still work, or what other physical insight can we learn, right? So far, I think we have produced a couple hundred embryos data now. Not full embryo, but only a slight, we'll take a regular fluorescent wide field scope. And Shaoxun developed the automatic algorithm to, segment the embryo and then trace the migration all automatically and convert it into a graph data structure that allow us to do what we want to do. So I think it's very cool, and I really look forward to this. All right, so to summarize, I really want to sell you this vision that, using AI in predicting protein structure is very popular in the last 10 years-ish. Part of the reason is because the protein data bank is very mature. There are a lot of data about protein structures, right? And last year, there are a couple major groups or associations starts to propose a virtual cell idea based on multi-omics, proteomics, transcriptomics, and can they develop a foundation model to start to make predictions of how each individual cell responded to different variations, right? What's essentially missing, I think, is in the MultiCell field, where we have many, many cells compose the tissue. Can we also build a foundational model there to start to predict what's going on in the tissue level behaviors? So I hope the MultiCell that we developed is one of the important works there, but there are a long way to go. And one of the major issues in here, as compared to the protein, the single cell, is the data and the quality and amount. We really need a lot of good quality data in a consistent way that help us to train the model. It's not easy. So I want to call for collaboration. If you're interested in machine learning, that's great. Come talk to us. If you have interesting, living systems that you want to image, we're happy to use our confocal. If you don't have one to image your system, we just want to try our model on yours. I think there's a bright future in this field. With that, I just want to acknowledge my group, collaborators, and the funding. The picture was taken last year, so some of you are not on it. I apologize. With that, thank you, and happy to answer questions.
This is just amazing. Okay, I'll give time to audience first. Go ahead. George, wait. I need to pass this mic.
Okay, yes, thank you, Ming. That was an awesome talk, and of course I was expecting no less. I have a question about the first thing that Hao Qiang did, this sort of phase change from what do you call it, to jamming, right? Where the last phase is jamming. So I was looking at these videos and I was thinking there's got to be hierarchical interactions. Yeah, that one. Sort of near and further away, because this is an elastic system, right? So and I was thinking, well, a power law has to come out, and lo and behold, indeed, what you saw there are power laws, right? So I was wondering, what do these powers tell you? Is there a way to extract physical information about the slope, from the slope of these curves? And maybe that's a way to regularize machine learning, because, if the slope is meaningful, it means that something correct has been learned.
Yeah. Excellent question, George. I think it's really, worth to think about. First of all,
Sorry, I think the next plot actually, it was even more evident. Yeah, this
right, exactly.
Look at that, right? Yeah.
Yeah, normally, having a power law suggests there's no characteristics in the time or the value, because it's a power law, right? But the power itself, indeed, can be quite important. I haven't thought about it, but I think it's good to talk more. Yeah, I think it's a great question.
I owe you a lunch at some point.
Yeah.
Great. Go ahead.
Sorry, I can see that. Thanks for a great presentation. I have 3 questions about, and the first question about the experimental. I just wondering much time, because how does cell perceive the mechanics by the cell nuclear, or by the cell memory? I want to know your opinion. And the second question about the about just like this. To the epithelium, because as I know in the recent paper, that they found when the cell, just like the scale recover, the epithelial cells will be restrained, and will some cell will be round and dead.
Yep.
So so maybe I think this phase will be influence your prediction model.
Yeah.
If you have concern this.
Yeah, great question. So going back to your first question, how they, will perceive or sense the mechanics, right? So there are a couple of different parts. One is the nucleus, and also there's a membrane. The membrane is the, outer boundary of the cell, right? That's where they're touching the environment, touching their neighbors. So when the rigidity of the membrane change, you can imagine the interaction will start to change. And another thing is you have a lot of proteins on the membrane, right? When the rigidity or fluidity of the membrane changes, the protein dynamics will change. So that's one way that they can sense that. And for the nucleus and cytoskeleton, I think that's even more direct. For example, this nucleus, if I apply a mechanical force on it, depending on its mechanics, it's going to deform differently, right? If it deforms more, then it certainly will influence the genome because you're stretching out the chromosome, you're changing the gene expression. If they're stiffer, so they deform less, or if they have the protection from the intermediate filament, they deform less. So then they certainly sense the mechanical force that way, right? And with their mechanics directly involved in the sensing. Going back to your second question, is the cell extrusion. That happens a lot. I don't think it will change the prediction. If the data is in the training set, I think it should also learn when that happens. For the embryo case, cells imaginate into 3D, right? That's very similar to a apoptosis, the cells being squeezed out, for example. So I don't think that will be an issue. Yeah, if the data is there.
Thank you.
Sure.
More questions?
Here.
Okay, I can ask one, but I think that was probably make very interesting talk. So I want to go back to the curved surfaces and the velocity correlation. Certainly the correlation decreased at the beginning, but for many cases it appears the correlation comes back up again. So I was just wondering, do you have some comments on that? And why does the correlation seem to increase again?
Yeah. So I say if you look at some of the curved surface, it becomes a little more clear, right? They decrease to an active value, suggesting they can reach anti-correlated state, right? And outside of this anti-correlation, they become positive again or slowly decay to zero. So it's not really increasing. It's actually because the anti-correlation is very strong, right? So from the negative correlation to no correlation at all, which is at zero, you have to increase back, right? It's purely due to the swirling, I think. Yeah. I think Matthias had a question.
First.
Oh, sure.
Great talk, Ming. I'm always looking for mechanisms in the data that you show, and I'm wondering, is there any way that you've been able to identify where you can start to say whether the signaling between cells is through, say, mechanics or through biochemical signaling or electrical signaling between cells? Any in terms of like either the length scales that you see the cells communicating over, or the rate at which signals or communication drops off?
Right. It's a great question. So I think depends on which system, but mechanical correlation and correlations through chemical definitely are involved in, I think, in all of those actually. The mechanical correlation as a clear example, as one cell deform, you can look at the deformation field if you calculate, right? Often it can span through a couple cell diameter. So in other work that I did not talk is you can start to look at the area fluctuation, their distribution, and through which you can directly compute with how many neighbors each cell is mechanically communicating. I can send you the draft is under review right now. So, I think that's a good way. Basically, based on how the area is exchanging from one to its neighbor. I actually think that's a really important thing for the graph, for the machine learning part, because for machine learning, you use multiple layers of the neural network, right? Each layer, you start to convolve one layer of neighbors. The more layers you have, the more neighbors you have. So you can start to consider a long-range interaction, which is not immediately, visible if we look at the data by eye, right? So I think that's a benefit also.
I mean, super impressive to see how this all comes together, both on the experiments and on the modeling, right? You have full stories to tell about all the different things that cells do in the mechanics. The AI model that Haichang is doing is super interesting because it learns on what it sees, right? You just see spatial information, the boundaries of the cells and the cell positions. But what tries itself from now to in 5 minutes from now, it's got to be, as Roger said, the mechanical and the chemical interaction itself. So what does the model see that allows it to just on the spatial information be able to tell the mechanics and the chemistry that might be in there?
Right. That's a great question. It's something I really want to answer. I don't think it's an easy question. It's a very hard question, right? Currently, our graph neural network, all the data is entirely based on the physical parameters, right? But the physical parameters could include some of the genetics already, right? Because it may change the physical morphology of the cell. So I think it's heavily involved there. But I think using some of the genetic perturbation, I think we can combine with the graph neural network to selectively study, some of these pathways. I think only by that, we can start to learn that, I think.
Very cool. Thank you.
Last question.
So I had a question about the predicted versus the true value. So what were the red color and the other colors around the straight line?
Oh yeah, so while I flip to that, it's the density of the points, I think, or probability. Yeah, it's a probability density. So being highest here is a special way to plot the database.
And the follow-up question is, so you have thought about using graph neural, the dual graph thing, like considering both foam behavior and the other type of behavior. So have you thought about like something which is completely different from the two, like a new model with graphs that is not either like the two of them? So have you thought, so that it can improve the accuracy?
Yeah, well, I'm sure, he thought, or they thought about it, where they use the graph transformer, they use some of other large language model as well. I think there are a lot of, interesting possibilities for us to try. Yeah.
And
sure.
Great. In the interest of time, let's thank Ming again for this wonderful time.